Namaste children, this is Dr. GV here, Dr. Venkat from RN NEET. We are going to be presenting today on a very important topic, structural organization in animals. This chapter is going to be a complete full shot. We are going to be doing the part 1, which is basically about mainly the tissues, organs and organ systems. Frog and cockroach we will be covering in the subsequent aspect. Now this particular chapter, as you look at it, is very important because basic understanding of the tissues and the organs is very important for your subsequent topics, including human physiology and every other chapter, including animal kingdom. So we will quickly start this particular chapter on structural organization of the animals, and we will look at actually how the different tissues, organs are actually based at. This is a complete class 11 important chapter that we would want to go for. See, the animal tissues, if you look at it, could be variable depending upon whether it's a unicellular or a multicellular. Unicellular means there is a single cell, so onde cell irutte, so ella tara functions so that one single cell have to do. Multicellular means there are different types of cell, so some will take different speciality, and so therefore these cells needs to be grouped together. So they are a group of cells and they need to be grouped together. Now when you basically want to do the basic understanding of this chapter, you have to start from how do you define a cell. Now very important, these important words, idu pakka gnapaka itkobeku neevu. See, cells are structural and functional unit of life. So life has to go on, so you need a structure, you need a function. So cells andre adralli basic usme structure hota hai, function hota hai. When these two are put together, that is when you call it the basic functional unit of life. So that is what cells are. Now what is additionally important is there are different cells, like in a complete office or my hospital, if you might say, there are different departments. There is a reception department, there will be people involved in that, there will be accounts department, there will be people involved in optometry, there will be some people involved in checking, and of course there will be doctors like you, future doctors, and myself, who will basically be taking care of the major part. So here you have different group of people, each one of them having a different division of labor. Now that division of labor is a very important thing. Now why that division of labor occurs? Because they are specialized and differentiation. So due to differentiation and specialization. So these three important parts define a cell. So these words you should be remembering: structural unit, functional unit, division of labor, specialization and differentiation. Now what happens is when group of cells come together, you have a single cell, now you have a group of cell, another group of cell, another group of cell, all of them actually looking very similar, probably having the same origin, specific function, and held together. They're not just loose, they're not like they are kept separately loose. You look at them, they are actually held together. There will be some substance like a cement. If you have basically a bricks which are held together, and these are all held together, if you can see, by some kind of a substance. So this particular thing where they are held together by a substance becomes your intercellular matrix or extracellular material. And you call it extracellular or intercellular, both are okay. Only 4 basic types of tissues are present, and we are going to be studying this specifically in this particular chapter. Now the organ basically then, what is, if you know that this is a tissue, what then is an organ? Organ are basically tissues which are organized. Now cells grouped together, specific type of a function, okay, and held together by a cementing substance. Now if tissues are organized, multiple tissues are organized together in a specific proportion, so which means you have A tissue, B tissue, C tissue, 3 types of tissues, assuming they are all organized together in a specific proportion and pattern. There will be a pattern to it. Either, for example, if I take a simple tissue, like for example, you might call a lung. You will have different tissues there, but all the tissues that help you to inhale, the ones which have tube-like, are all put together. Then you have the smaller tubes. Then you have the air sacs. So there is a specific arrangement. So the arrangement is very specific. There is a specific proportion, and they together form again a specific function. So as tissues which are organized together for the sake of a specific function, arranged in specific proportion and a specific pattern, that becomes your organ. So it could be a simple things like stomach, a liver, urinary bladder, a gland, a salivary gland, eye. So these are all, every part of it becomes a organ. When you have organ system, the moment you say it's an organ system, it means there are not one organ but at least two organs. You need to have minimum of two organs to say it is an organ system. The purpose is that you have two organs, but they function together because of some interaction. There has to be an interaction between one tissue, one organ and another organ. So if there is a specific interaction between them, andre ಎರಡು ತರ organs ಇದೆ, ಒಟ್ಟಿಗೆ ಕೆಲಸ ಮಾಡಬೇಕು. Okay, so for example, if I want to say a simple two organs which actually work together, the heart and the lung. Correct? So when the heart pumps, basically the blood moves, comes to the lung, purifies, gets back to the heart. So these two keep working together. So in a coordinated manner, and they are governed by a specific signal. So even two organs, when they come together, interacting together physically, chemically in a coordinated manner to carry out the major life processes, and these are called organ system. So the cardiac system here consists of one, the heart and the blood vessels. So there are blood vessels bringing blood to the heart. There are blood vessels taking out blood from the heart. So it's a common thing where something is bringing, something is pushing, again something is taking it out. So there is a system. When number of organ systems bring together, work together in unison to sustain an individual, such a thing is called as an organism, and we are all organism showing the highest level of structural organization. So andre level tara some cells के बाद में बहुत सारे सेल्स एक ही तरह का काम करेंगे, टिश्यू. जब टिश्यू बहुत सारे सेल्स रहेंगे बट डिफरेंट टिश्यूज, बट उनका एक तरह का ऑर्डर रहेगा, पैटर्न रहेगा, एक तरह का सिमिलैरिटी रहेगा, वो ऑर्गन बन जाता है. और टू ऑर्गन्स ब्रिंगिंग टुगेदर, वर्किंग टुगेदर, एक तरह के पैटर्न में वर्क करते हैं, साथ में वर्क करते हैं, देन बिकम्स अ ऑर्गन सिस्टम. Organ systems, multiple organ systems becomes an organism. So this is the basic about how you define one to the other. We'll go to the next important part, which is basically your most important part that we want to go for is your animal tissues of the next portion, which you see here. For example, a simple thing like hydra. Now hydra is made up of different types of cells. Now is hydra having number of cells, number of tissues, number of organs, what kind of or level of organization it is? It is one kind of a animal tissue which has a tissue level of organization. Very important to remember this, because it has not just number of cells. These cells are all arranged in different tissues. So if you look at the hydra, it has a mouth, but that mouth is not like separated out. It is again a part of the same tissue. You have the tentacles. Again, the tentacle is a part of a tissue. Then you have, for example, a bud. You have the gut. You have the ovary. You have the testis. You look at it, they're all outpouchings of the same tissue. There is one tissue which is going from one end and then going around like that and then coming out on the other end. So this particular thing where it is moving from one to the other is typically called a tissue level of organization. There is a very classic important thing you need to remember here, that NCERT give these words that it has each cell type, the number of cells present in each type of tissue could be of the order of thousands. So remember this word, thousands in Hydra. Whereas if it is human body, it is composed of billions of cells. How many? Billions of cells which perform various functions. Now, there's a very important question basically that has been asked. Look at this basically, which tells you about which of them is correct. Different cellular level organizations they have given, different types of organizations of tissues or cells they have given, and they have asked which of this is correct. Sponges show cellular level of organization, Cnidaria tissue level of organization, Platyhelminthes organ level of organization, Annelid, arthropod, mollusks, echinodermata, all organ system level of organization. Which of this is correct? Put it in the comment section. See whether you are able to revise your animal kingdom. Now, very important is, see this particular thing. Understanding at animal kingdom directly is a difficult part because you would not understand what tissues, what is the role of it. But when you read this chapter and then do animal kingdom, you'll be able to register these things much better. So in this question, you see that sponges, yes, they are the basic portion which starts from sponges. So they show cellular level. Cnidaria, you saw that here, Cnidaria represented by Hydra has tissue level of organization. Platyhelminthes, all the flatworms, all the flatworms, okay, that are there, the tapeworm, the ascaris, all of them. So these show platyhelminthes, which shows organ level of organization. Annelids, arthropods, you automatically know they have various organs and they are already evolved creatures. So they have organ system. So here the answer is all of them are correct. Right? Perfect. How do you basically classify animal tissues? We said that we want to classify animal tissues primarily into 4 main types. What are the types? 1, epithelial tissue. Okay, epithelium basically means anything that is causing a covering. Ethelia basically, means epi means outer, and thelia actually doesn't really mean a cavity or anything. It is something basically stands for a surface which is actually covered externally or internally. It is covered. So that is what epithelial tissue means. Then you have muscular tissue. What do you mean by muscular? Muscular means anything wherever there is muscle. Connective tissue. The word connective itself means it is a tissue which is connecting one to the other. And nervous tissue. So 4 main tissues, simple: epithelium, muscular, connective and nervous. Under epithelium again you have different types which we are going to go in detail. Whether simple squamous epithelium, stratified squamous epithelium, basically squamous, columnar, cuboidal, and there is a variation of it called the glandular. So these are the 4 main types of epithelium. Then if you want to look at what are the types of muscular tissue, we will come to that, the different types of connective tissue, and of course the nervous tissue. So this gives you the broad kind of classification of what are the different animal tissues. We'll come to the most important first part, that is the epithelial tissue. Okay, now the most important part about epithelial tissue which causes a covering basically is either from outside or inside, is it has a free surface. Now the epithelium basically means there should be a free surface. Where is this surface? Either to the outside or facing to the inside. So which means either facing inside a cavity. So if you had your hydra, okay, you had tissues which are present here, but where are they facing? They're facing the body fluid inside the cavity. So if the tissue is facing inside the cavity, or the tissue is present on the outer surface facing the outside environment. So that is very important. So it is a tissue covering which faces either the outside environment or the internal environment, and that is a free surface. So automatically means one surface is free, so the other surface is attached. Where is it attached to? Invariably there will be a basement membrane, a kind of a sheet on which these cells will be sticking to each other. And what is their purpose? They provide a covering or a lining. They provide a covering or a lining. Okay, ಮೇಲಿಂದ ಒಂದು sheet ತರ cover ಆಗುತ್ತೆ. And they are compactly packed cells. Okay, they are compactly packed cells with little intercellular material. That means there is some material which is present in between the cells, but that material is very less. It's a scarce amount of material. So this is how you basically classify. And if you look at basically the different cells, the cells can be either squamous. Okay, you look at it single layer. If it is little bigger, then they are cube-like. This is cuboidal. Or they could be long, big column-like. So therefore they are called as columnar cells. So basically three types: squamous, cuboidal, and columnar. If you look at basically what are the then what is the other part? If the squamous is present as a single layer, or the squamous is present as a multiple layer. If they are present as a multiple layer, it becomes stratified. Cuboidal again, simple single layer or multiple layer. Similarly, when you have columnar, again simple, stratified, and there is something called pseudo-stratified. In pseudo-stratified, if you look at it, they are not two layer. If you look at it, they are not two layers, whereas they are just one single layer, but the cells are such that some of them are large, some of them are in a different level. So you will see that some of these cells are little bit bigger and in some of them are little smaller, and the nucleus is present at different levels. So if you look at it, they are each single cells but at different levels. So that's why that is called as a pseudo stratified, because it looks like as though they are multiple layers, but they are only single layer. Okay, now let's look at basically the squamous epithelium. Now in all these things, one of the key things that you need to remember is where this epithelium is. ये epithelium कहाँ होता है, उसका function क्या होता है, और what is this, any peculiarity about that feature. So this is the way you need to remember this. What is it, what is its function, and where is this particular present, and how I can remember them. Okay, we'll give some tricks as you can go through. Most of it will come by repetitive practice. Okay, squamous epithelium. It is made up of a single thin layer of flattened cells. That means cells are all very flat. Okay, and very important is the irregular boundaries. The boundaries are not regular. Okay, so there will be some kinds of cells here, okay, layer like that, but the boundary is not always regular. See, here it is straight, here it is irregular, again it becomes straight, again becomes irregular. So they can be irregular boundaries, very important. Okay, and found in the walls of the blood vessels and the air sacs. The most important are the walls of the blood vessels, the air sacs, okay, and single layer. They are single layer. Okay, skin also is epithelium, but I'll tell you, it is not a simple squamous epithelium. It is a stratified. There are different layers in the skin, whereas simple layer is, single layer is, the alveolar sac. So the sacs are there, and within that you have a single layer of cells like that. Okay, and then similar kind of thing is what you get in reed testis. You would have probably read in reproduction in class 12th, those who are basically revising. 12th standard students who are now revising this will understand that how basically reed testis consists of different kind of areas or round cavities with a single layer of cells which are covering them. So that is again a simple epithelium which is covering them. Another very simple structure is in the kidney. Okay, the kidney basically you have the Bowman's membrane, and this Bowman's membrane consists of a single layer of epithelial cells. So this is again a simple squamous epithelium. Correct? So air sacs, inner lining of blood vessels, again the blood vessels are there, and the inner lining you have a single layer of cells on either sides. The ones that are lining the blood vessels are called as endothelium. Now they are not called epithelium, they are called endothelium. Okay, now this is important because they are simple. Anything outside is epi, anything inside is endo. Okay, and then what is another important feature you need to know? What is their main function? Because they are single layer, obviously they cannot be for maintaining thickness or strength or anything, because there you need more layers. They are simple single layer because they will allow passage. You look at it, air sacs allowing passage of oxygen and carbon dioxide on either side. Bowman's membrane allowing passage of all the metabolites, urine, filtration that takes place. Okay, reed testis, wherein you need to have transmission of the spermatozoa, and so therefore these are also single layer. So every particular portion, including the blood vessels, where there needs to be transfer of oxygen, transfer of nutrients across the blood vessel into the tissues. So again there it needs to be a single layer. So wherever there is a diffusion, okay, ಒಂದು ಕಡೆಯಿಂದ ಇನ್ನೊಂದು ಕಡೆ ಸಿಂಪಲ್ ಡಿಫ್ಯೂಷನ್ ಆಗಬೇಕು ಅಂದರೆ ಒಂದು ಮೆಂಬ್ರೇನ್ ಇರುತ್ತೆ, ಆ ಮೆಂಬ್ರೇನ್ ಇಂದ ಒಂದು ಕಡೆಯಿಂದ ಫ್ಲೂಯಿಡ್ ಇನ್ನೊಂದು ಕಡೆ ಈಸಿಯಾಗಿ ಹೋಗುತ್ತೆ. ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಡಿಫ್ಯೂಷನ್. ಸೊ ವೆನ್ ದಿಸ್ ಥಿಂಗ್ ಹ್ಯಾಪನ್ಸ್ ವಿಚ್ ಟೇಕ್ಸ್ ಪ್ಲೇಸ್ ಅಕ್ರಾಸ್ ದಿ concentration gradient, you know that if the concentration is more here, then in that case the tissue fluid from one side will go to the other side from a higher concentration to a lower concentration, right? So that is diffusion boundary. The other important characteristic feature of these cells are they show regeneration. That means they can regenerate. They are avascular. Now ಏನಾಗುತ್ತೆ, because they are already ಒಂದೇ layer and they are maintaining diffusion, they will derive their oxygen through diffusion itself. When oxygen passes through them from one layer to another place, automatically the oxygen can be utilized by these cells, and so therefore they are avascular. And the important feature is there is a basement membrane. So the cells have to be remaining there, and they need to be sticking on a sheet of membrane, and so that membrane is a basement membrane. So you got the whole idea how to remember these things? Simple. So simple layer, single layer, simple squamous. Multiple layers, stratified squamous. Okay, so plus they have to have irregular boundaries. First point. Second point, present in blood vessels and air sacs, most important. Along with that, reed testis and Bowman's membrane, diffusion boundary, regeneration, avascular, basement membrane. So very important points. ಈ ಥರ ನೀವು ಒಂದ್ಸತಿ ಕೇಳ್ಕೊಂಡ್ಬಿಟ್ರೆ next time you will never forget, and you will know how to go categorically and how to remember each of these points. But ಈ ಒಂದೊಂದು ವರ್ಡ್ಸ್ ಟ್ವಿಸ್ಟ್ ಮಾಡಿಬಿಟ್ಟು ನಿಮಗೆ ನೀಟ್ ಕ್ವೆಶನ್ಸ್ ಅಲ್ಲಿ ಕೇಳ್ತಾರೆ. ಸೊ ದಟ್ ಇಸ್ ದ ವೇ ಪೀಪಲ್ ಗೋ ಬೈ. ಓಕೆ, fine. Now we'll go to the next one. We saw basically the simple squamous epithelium. Cuboidal. Cuboidal andre we know that, we told that they are clean cube like. Cube is what? All sides are equal. And so therefore cube like andre the nucleus automatically becomes in the center. So nice brick layer arrangement. Okay, so this is what basically happens. Now the beautiful thing about these cells is, they are single cube like, But what happens to these cells? On their apex, ಮೇಲ್ಗಡೆ, the cells are little folded like that. You people can see that. So this one particular cell, if I take, the top of that cell is not straight, it is folded like that. Okay, and so therefore these foldings of the cell, this is called the apical surface, that means on the top. The apical surface shows these multiple bending, and so they are called as microvilli. And these microvilli create a kind of an appearance like as though they are having a brush at the tip. They appear like small brush at that area, and so that's why they are called as brush border cells. Okay, brush border cells. And so therefore, what is their main function here? Simple. Again, they are single layer. They cannot be for only diffusion because the surface is folded. So something should extra happen from these cells. What should happen? They should be meant for secretion. Their major function is secretion or absorption or excretion. So secretion, absorption or excretion. So remember these, that it has to be because microvilli are there, it should be to allow passage of something. So it should either secrete or it should excrete or it should absorb. It should either take into the cell or give out from the cell. Simple as that. So this folding is basically done so as to increase the surface area. You know that if the cell has only so much space, now if you put it more curved, obviously the surface area is increased. So that is why more amount of absorption can take place. So very simple, you know this structure. Obviously kidney, tubules, your proximal convoluted tubules, your distal convoluted tubules, all of these should have cuboidal epithelium. So now you will never forget that kidney tubules are cuboidal. Otherwise, very often this is something which is made a mistake and people tend to make a mistake. इल्ली तब माड़े मारता रहे, बहुत लोग इसमें गलत करते हैं कि PCT, DCT, PCT में cuboidal है, but DCT में cuboidal है या columnar है. कहीं पे if they give you a twist there and ask you that DCT में distal convoluted tubule में columnar है, तो कभी कभी हम मार खा जाते हैं, we forget it and we put that, you know, that may be right. But you should remember that for absorption, all this, be it the proximal convoluted tubules or the distal convoluted tubules, it needs to be the same cuboidal cells. Another important part is where you want these are basically not only the nephrons but also in the seminiferous tubules, because it has to secrete. The ovaries, the thyroid, very important. Okay, if you look at the thyroid follicle, the whole thing is a follicle, but all around them there are cubical cells, and these cubical cells secrete right inside the cells. Sweat glands, obviously some portions of the sweat glands will be cuboidal. Okay, so remember these main features which are actually going to be easily understood the moment you know that what is the purpose of these cuboidal cells. We'll come to the columnar epithelium. The columnar epithelium is what? Wherein the cells are more column-like. So they are large like this, column-like, big, and so therefore their nucleus tends to be aggregated more at the bottom. Okay, and their nuclei are located at the base and they are tall, slender cells. Surface have again microvilli. So multiple microvilli here, which basically allows absorption. And so these needs to be present for both secretion, and they are tall columnar because they need lot of material has to be secreted. So that is why all the glands involving the stomach, the intestine, bile duct, gallbladder are all columnar. Now very important point you should know here, while most of the GIT has columnar, okay, pancreatic duct is cuboidal. Pancreatic duct is very special in that the duct of the pancreas is not columnar, doesn't secrete cells like the other ducts, it is cuboidal. So remember this one exception, the pancreatic duct is cuboidal. Automatically you will remember that bile duct or GIT, stomach, duodenum, other parts are all columnar and their major function is to secrete and absorb. Okay, so this is a very important aspect about it. Okay, we come to the next important part, which is called as a ciliated epithelium. Now, what is the ciliated epithelium? Is it something different from the squamous, columnar or cuboidal? It is different, but how different? Simple. It is either a cuboidal cell or a columnar cell, and this particular cell, apart from having microvilli, has thin hair-like structures. They have hair-like structures, and so therefore they are cuboidal, or you can say columnar. What is their purpose? To allow movement of particles, okay, in a specific direction, or mucus. थिक म्यूकस जो है ना नाक में जो आता है म्यूसिन आता है कोल्ड हो जाता है तो म्यूसिन आता है so that is the mucus which is there and this mucin what is there you don't want it to go and block somewhere so it should either come out in only one way or similarly you don't want dust particles to enter inside. You want it to be thrown out. If supposing dust particles are inhaled inside and creates a choke, so your trachea, your bronchi, all of them have got this ciliated epithelium and they allow the passage of these particular dust particles outside. They allow you to cough it out and so therefore that's a protective mechanism. So therefore you want them to move the particles in a one-way direction and not in the other way. Okay, and they are present on the inner surface of hollow organs like the bronchioles and the fallopian tubule. So the air sacs, the nasal cavity as well as the bronchioles and the fallopian tubules. Fallopian tubules, the purpose is it needs to move the ovum so that it basically moves from the fimbriae to basically the place where the fertilization takes place. So it's very important that along the tract of the UTI that basically from the fimbriae it moves around from the ampulla and then reaches basically near the isthmus. So it's very important that the fallopian tubules also should have the ciliated columnar like the bronchioles. Okay, so you understood why the ciliated epithelium is important in these parts. We'll look at this another part which is called the glandular epithelium. Is it any different again? It is nothing but the word speaks about it. It is a gland. So it is an epithelium of a gland. So that means something is secreting out of it. Now it is very similar to a columnar cell. Okay. Very important here is it could be either a unicellular or a multicellular. Multiple cells together can become a gland or a single cell can become a gland. If it is a single cell, very important example of this is a goblet cell. Okay, this particular thing is a goblet cell. Okay, it is in that kind of a triangle kind of a shape. Now, what does the goblet actually means? It is also called as a callus cell. This thing is hardly told anywhere. But okay, there have been questions which has asked what is a callus cell. So that has been one of the questions that has been asked. So what is a callus cell? A callus is, like your, the British people would sometimes drink wine. It's called for the victory cup. And so therefore it's like your victory cup, okay, having a broad top and a narrow base. A narrow base and a broad top. So this particular thing like a cup which is actually called the callus. Okay, now this callus is what is represented by this goblet cell. So a narrow border, bottom, a top is more, and then microvilli in the top, and then the nucleus is at the bottom. So very similar to columnar, except they are not tube-like. The bottom is narrow. So that is called as a callus cell or a goblet cell. And where are they present? All throughout your alimentary canal. Where you basically have to swallow, your alimentary canal is called the callus cell. And they can also be present in the conjunctiva. What is conjunctiva? The outer layer of your eyes. That is called the conjunctiva. And so there also mucin is secreted. So the major function of goblet cell is to secrete mucus or mucin, which gives you smooth passage. You don't want the food to choke, you don't want it to irritate, so you need a smooth covering throughout your alimentary canal, and so that is what mucin does. Multicellular layers sometimes can be present, like the salivary gland, the sweat gland. Okay, all of these give you, or those giving you digestive juices. So the salivary glands, sweat glands are all multicellular, and when they are multicellular, they can basically be of different shapes, and they can either secrete into a single duct or multiple ducts. So that way each one of them are either simple or compound. Okay, so this gives you the idea what is a glandular epithelium. When we read more questions, we will come to them as to see how different each one of them, how different they are. Okay. Now, glands are either divided, into exocrine and endocrine. What is the exocrine? An exocrine is one where it secretes something, like for example mucus, saliva, earwax, oil, milk, digestive juices. So all these are all secretory products from either mucus, the saliva, the earwax, the oil, the sebum, the milk, and the digestive juices. So all of these are secretory products, and they are secreted through ducts or tubes. If they come out through tubes, that is called an exocrine gland. The same secretion doesn't occur through ducts, but directly the secretions enter into the blood, okay, or enter into a fluid without a duct, into a fluid which is bathing the gland. Such a thing is called a endocrine gland, and their major function is hormone. Okay, so you got this basic difference between an exocrine and an endocrine. Okay, now what is a merocrine or a mixocrine or a heterocrine gland? There is something called a mixocrine or a heterocrine gland. What is this? Any idea? Put it in the comments. I would like to know how many of you know this. What is a mixocrine or a heterocrine gland? There is only one gland in our body, which is this. Can you put it in the comments box? Fantastic. Great. As many of you who have been able to put it, great. We will review it subsequently. Meanwhile, don't forget to subscribe, like, and follow, because we're going to ensure that your understanding of especially biology is too good. At the same time, we're going to be ensuring that your main concepts in chemistry and physics are also taken care of. So the idea is to ensure that you get that 360 out of 360 in biology. Perfect. And we're going to then go and strengthen your physics and chemistry and see to it that you maximize your potential in these two subjects. So what is a heterocrine or a mixocrine gland? It is one which behaves both as an endo and an exocrine gland. Which gland is that? The pancreas. The pancreas is the only gland which is both exocrine and endocrine. It produces insulin, it produces digestive juices. Digestive juices come out through the duct, so it's partly exocrine. Whereas insulin, glucagon, which are produced by the pancreas, again is endocrine. So that way it's called as a heterocrine or a mixocrine. It's a mix, okay. Now we come to the other part, which is called the pseudostratified. Now we saw that like sometimes you can have different layers in the tissues, but there is one particular kind of cell which doesn't really have different layers, but it appears like having one. That is called the pseudostratified. The cells are all single, but you see some cells are small, some cells are big, so resulting in a varied arrangement where it looks like the nucleus of the different cells are in different levels, causing it to appear like as though they are in multiple layers. This can be either a columnar or it could be ciliated. If they are columnar like how it is here without any cilia, it is mainly present in the parotid gland. Parotid gland, which is basically the gland present here, which causes swelling, then it causes what is called as mumps. Okay, so the swelling of that gland is called the parotid gland, and the parotid gland is one gland which is columnar pseudostratified. Another very important part you want to remember is the olfactory mucosa. What is this? The mucus involving your breathing, the olfactory mucosa. Olfactory means your olfactory apparatus is this particular portion which takes your smell. Okay, when you breathe in through the nose, the upper portion top, underneath the upper portion of the mucosa, you have the mucosa which basically helps you to detect smell. And so that particular mucosa is actually pseudostratified. Similarly, the urethra in male, very important part, you should remember this, because urethra in male has got dual functions, One, is excretion, and the other one is also reproduction, where it basically has to transport semen. So you want it to transport both urine and semen. So that is why this particular tissue has to be specialized and is not stratified but pseudo stratified. So you try to figure out some reason to understand why this should be behaving something differently. Okay, so that's why urethra becomes a pseudo stratified. Parotid we said, and the olfactory mucosa. Ciliated. Similarly, you can have pseudo stratified and ciliated also. Now the smaller bronchioles we saw and we told that they are columnar ciliate, right? But the larger bronchi as well as the trachea, if you have those cells as a single layer and not very thickened and not variable, they're larger structures. So therefore, if they have a very small cell or a slender cell, it could be dangerous. So therefore, in order to give it extra protection and make it more, thicker, the epithelium of these tissues, especially the trachea and the bronchi, is ensured that they are made thicker with the pseudo-stratified appearance, right? Perfect. Now we come to the next part, which is called the compound epithelium, made up of more than one layer. Now, if automatically there is more than one layer, and if each of those layers appear either similar or basically same, then it is a compound epithelium. Obviously, its compound epithelium will have a limited role in secretion and absorption. You know that only those layers which are single layered have a greater ability to absorb or secrete, and so therefore compound epithelium function is not mainly for excretion or secretion, not that it cannot do, but limited role. So then what is the major role? To provide protection. What does it do? Ontara, it's like a thick multiple layers. So it's like a brick wall. So ontara protection kodatte. It will prevent stress, both mechanical, adu ಏನಾದರೂ ಒಡೆದೋಗ್ಬಾರ್ದು ಅಥವಾ ಕೆಮಿಕಲ್ ಡ್ಯಾಮೇಜ್ ಆಗಬಾರ್ದು ಅಂತ ಇರುತ್ತೆ. So that is why it gives you a more amount of a protection. Okay, and what is the major another part of it? Example you can see is simple as skin. Okay, covering the external surface, the dry surface of the skin, because again there are multiple layers: epidermis, dermis, you have different types of layers, cuticle, basement membrane, and so therefore each of these, the external surface of the skin, the moist surface of the buccal cavity or your mouth cavity, the pharynx, the upper portion, the inner lining of the ducts of the salivary glands. So all from your mouth you start, okay, the buccal cavity, the pharynx, the salivary glands, okay, and very important, as I told you, the pancreatic duct. Pancreatic duct is one type of an epithelium which is compound and is cuboidal. It is not columnar, it is cuboidal. We told that earlier. We'll go to the next part, which is basically stratified squamous. We saw that compound epithelium, number of layers. In the first part, stratified squamous. You know what is squamous? Single layer, flattened cells. Same thing, multiple layer, flattened cells. That is called stratified squamous. You know that obviously skin is one thing where there is stratified squamous. The same thing, if I look at it, basically is, if you want to basically look at what is in the skin, there are certain portions which are very hard. Certain portions of our skin is very hard, or even the nail is extremely hard, so the nail bed is very different from that of the smooth skin. So part of the skin could be what is called as keratinized. Keratin means thick kind of horny material which is secreted, which makes it very, very hard. Under your toes, if you see the callus that is formed, that is all very thick. The same skin which is there, in another area is very soft, whereas certain areas is very hard. So the skin which is actually present at the callus, the base of your feet or the toe, is basically the keratinized, and their role is basically to check water loss. Okay, so keratin means it's a thick material which is present, and that material ensures that water cannot pass through, so prevents water loss, and also for giving great amount of protection against any damage, any tissue. So the feet, underlying of the feet, that's why needs to be very hard, so that it can prevent, even if a stone hits your skin, it should not damage your skin frequently. Infection could not get in, so therefore protects you against abrasion and infection. Similarly, the non-keratinized is basically does give you protection, but the amount of protection is lesser. That is why all the mucosa, as we said, the pharynx, all of that, including the vagina and the cervix and the cornea and the conjunctiva, are all non-keratinized. So easy for you to differentiate where it should be keratinized or not keratinized. Okay, now stratified. Under the stratified, we also said that some of them could be columnar, multiple layers of columnar cells. Where are they present? Remember the mammary glands. This is a very commonly asked question, important. Okay, the mammary glands, the urethra, and the epiglottis. Epiglottis, the one which covers your larynx, that particular structure, okay, which prevents food from entering your larynx, is your epiglottis, and that structure has stratified columnar cells, okay, along with your mammary glands and urethra. Ciliated columnar stratified, ciliated because you don't want dust to enter into the air cavity, so your larynx has got ciliated. Non-ciliated, the mammary glands and the urethra. Stratified cuboidal. Cuboidal cells which are present in multiple layers, typically the pancreatic duct, salivary glands, some areas of sweat glands. Mainly you need to remember pancreatic duct and the female urethra. Male urethra has got stratified columnar, whereas certain areas of the male urethra has to be stratified and has to be columnar, whereas the same thing in females, it has to be stratified cuboidal. So the female urethra is stratified cuboidal, male urethra is stratified columnar. Then there is a very interesting epithelium called the transitional epithelium. What is this transitional epithelium? Can you put it in the comment box and let me know as to how many of you know what is this transitional epithelium and what is the speciality of this transitional epithelium. Let's look at that. See, the transitional epithelium is called transitional epithelium because, there is a transition in the epithelium. That means it is some portion of it is flat, some portion as they grow up, the cell layer changes from becoming initially some cells of it basically being, flattened. Some cells on the top layer becomes more like umbrella and more bulgy. Okay, like small dome shaped cells they become. So the cells change their shape from the lower layer to the upper layer. They are stratified, but they are present in different shapes. So the bottom layers, okay, at the top bottommost portion of it, okay, they are called the basal layers, and these do not show mitosis. No mitosis in the bottom layers, 4 layers. So this particular layer do not show mitosis, they don't multiply. Then you have the above that the columnar cells. So the basic cells are there which don't show any kind of multiplication. Over that you get columnar cells. These will multiply. The basal cells don't multiply. So the basal cells don't multiply. The upper cells multiply. Then you get superficial cells which are umbrella shaped or globular. So you see they are more like globe, like small bull, round features, and so they are like called globules, okay, like fat globules, and these are called the globular epithelium. Now the main reason for them being in different layers is that it allows a great amount of stretchability. It can distend, it can expand, and so that is why your urinary bladder, as more and more urine gets accumulated, it is able to stretch itself so much. The muscle also stretches, the epithelium inside also has to stretch and accommodate, and so therefore it needs a high amount of stretchability. So stretchability, distension and protection is the key word for transitional epithelium. Such an epithelium is basically the main epithelium of the urogenital tract, the whole urogenital tract from the bladder to the ureters, okay, and almost to certain portions of the urethra are all transitional epithelium. Fantastic. We come to the next important part, basically the junctions. What are the different types of junctions? And then we will go into certain other aspects, like for example the connective tissue. Okay, before we get into connective tissue, we'll take a short break. 5 minutes break you can probably take in and come back subsequently, but we'll then go ahead, finish what is called this junctions, and then come back for the next part, which is the connective tissue. So what are the junctions? When you know that there are different types of cells, right? Now you know that some amount of communication needs to keep happening, chemicals needs to move, sodium, potassium, minerals, all these needs to move from one cell to the other cell. And at the same time, you know that these cells have some intercellular substance which basically helps to keep them cemented. That means they are attached to each other by a substance. So while they are attached to these substance, there are also certain junctions called three main junctions. What are these junctions? One, the first most is called the tight junction. What is a tight junction? That means the cells are so tightly bound to each other that you can't separate them. No amount of material can pass through these junctions, and their purpose basically is to kind of a seal. You're like putting an M seal. Okay, remember that you're putting something like a sealing material so that no movement takes place, there is no leakage of molecules between them, and such a junction is called the tight junction. They are typically present at the top of the cell. This particular thing is one cell, this is another cell, this is the third cell. So there is one cell here and there is another cell here. The top of these cells here has a junction next to each other. So at every top of the junction, you have a tight junction, which is present at the apical surface. Apical means the top portion which are sitting to each other. So it's like a cement which is keeping them together and nothing passes between them. So this is called as a tight junction. The next important junction is called the adherent junction. That means it is sticky. They are adherent. They are touching each other. Something is there which is keeping the cells together, but they are not so tight. Some amount of molecules can move between them. Okay, but it is not that everything can move between them. The purpose of this is to create them from allowing them to stick to each other. So this can be called as adherent junction. One such junction is one which is formed with actin. You have read about the muscles, actin and the myosin. So similarly, you have actin filaments which bridge across these junctions, and such a junction is called as an adherent junction. It is basically meant to create an adhesion from one cell to the other cell. So these are called the adherent junction, okay, or zonula occludens. Then you have the other type of adherent junctions. The adherent junctions are three types. One is the zona occludens. So you call these as zona occludens. Zona occludens. The next one is desmosome. So what are desmosome? Here you don't have actin. Actin is not present. If actin is present, it is called zona occludens. If desmosome, that is basically intermediate filaments, are present, these are another group of filaments called intermediate filaments, and they are present from one cell to the other cell, and they are present not at the top, just below, not below the tight junctions, but in between the cell, in the middle portion. They are called the desmosome. Then there are third type of again adherent junction called the hemidesmosome. What are they present? They are also present not in between the cell, but between the cell and the basement membrane. Between the cell and the bottom portion, that is the basement membrane. So between the cell, here top tight junction, then zona occludens, and then you can get the desmosome, and then down here between the cell and the basement membrane you have the hemidesmosome. So here you have the hemidesmosome, and what is their purpose? To anchor the cell not next to the adjoining cell, but to the basement membrane, to the membrane which is sticking at the bottom. So there is one cell here, one cell here, one cell here, stuck to the bottom is the hemidesmosome. So simple. So top tight junction, then the zona occludens, then the desmosome, bottommost to the basement membrane hemidesmosome. In between at the bottom of the cell, you want a place where maximum amount of material can pass through, and so therefore such a junction is called the gap junction, which allows the passage of material to pass from one portion of the cell to the other cell. Okay, so we're going a little slow here. You can always speed up, go into speed or 1.5 speed basically to pick up in case you know these things very well. The purpose of this is basically to ensure that you read this once and you then have to just revise it once more later. You'll be so thorough in the concepts that you don't have to even turn the pages subsequently, because we are going to be understanding every portion of small things as to why something is called this, what is the reason for something to be like this, and what is the purpose, and how I can actually remember this rather than just memorize this. Okay, so that is the basic purpose how we will be going through. So you all understood that different junctions, basically three types: the tight junction, the adherent junctions, and then the gap junctions. Gap junctions will allow fluid. Tight junction is like a cement. Tight junction is like a cement. It will prevent passage. Adherent junction are different, either actin, desmosome or hemidesmosome. Basically it helps to just keep the cell next to each other, join and keep the cell next to each other. Perfect. So simple characteristic important words that are important here. We will then move on to the connective tissue. Basically, I'll take a short break and just get back to you right away. Perfect. See you soon.